Thank you, Richard, for the kind introduction, and thank you for the invitation. I'm really glad to be here and to be back to Hong Kong again and meet all of our colleagues and friends and discuss our most exciting science. And today I'm going to share you what we have done. This is a lot of the work over the past 1 or 2 years, focusing on RNA sequencing. So we're going to talk about RNA. RNA is, as we know from the central dogma, is the first thing that occurs after DNA transcription occurs. We got the primary RNA transcript, and then there are modifications, including the 5' capping, RNA splicing, et cetera, and we get a messenger RNA, and that got translated into protein. So why are we studying RNA? Why are we discussing RNA today? As you may know, that I've been working on the diagnosis of genetic disorders over the past 15 years. I have been focusing on DNA because that's where the answer lies, right? And the reason for that is we are looking for the molecular diagnosis for these patients with Mendelian disorders. And in the process of that, we are doing many of the variant curation. And we have learned that there are many important pieces of information that we need to obtain in the process of variant interpretation. And despite the advancements in the field, we really needed some more evidence to help us to improve the genetic diagnosis, to improve the accuracy of variant interpretation. So RNA sequencing comes into play as a high throughput technology that you can use, you can implement in a diagnostic laboratory, and then you get a functional consequence of the genetic variant that you obtain from patients that you cannot easily interpret. So that's the point on the molecular diagnosis part. We really need RNA sequencing to help us improve our interpretation of genetic data. So on the other side, we are also starting to work on therapeutics of genetic disorders. I would have never imagined that I'll be working on genetic therapeutics a couple of years ago, as we were just looking at the DNA. But actually, some of the new technologies made this a reality, including the advancements in RNA-based therapeutics. And one of the most common examples, as you know, is the COVID vaccine, right? That is a relatively different way of using RNA as a therapeutic agent. But the concept is that we can just look at the sequence and design some very specific targeted therapeutic options, and that may actually be helpful to treat genetic disorders. So I'm going to talk about our what we learned about RNA sequencing in the diagnostic part first, and then in the latter part of the presentation, I'll discuss the therapeutic developments we have done. So some of the basic concepts so that we lay the foundation of our understanding. There are, before you start processing RNA sequencing, you have to first make the library. And there are some of the things you need to take care of in preparing the RNA library. One of them is that there are a lot of ribosome RNA in the transcriptome that account for 80 to 90% of the sequencing yield, right? If you want to make the best use of your transcriptome sequencing data, you have to get rid of them. So there are commonly two ways employed to enrich or deplete the unnecessary materials. One of them is this poly-A enrichment, where we use probes to extract the messenger RNA, and we know that they're all modified by a poly-A tail. If you can enrich them, you are enriching the part of the transcriptome that you can most readily interpret, and you get the highest yield of data interpretation from RNA sequencing. The other way is you can decide to deplete the ribosome RNA. As we know, they are taking a lot of the sequencing yield if you didn't deplete them, and there are different ways, such as RNase-H, CRISPR-Cas9 depletion methods. And all of this, again, is to help you enrich the interpretable sequence from the transcriptome. Here I'm showing you a couple of representative papers using RNA-seq to improve molecular diagnosis. And you can see that diagnostic rates have been improved from 7% all the way to 30, 40%. So it has been demonstrated to be a useful technology. And some of the common modalities used in RNA sequencing interpretation are outlier analysis. So essentially, we're looking for outliers. For example, some of the genes are expressed much lower or higher than controls. So they are some good evidence to indicate that those may be the diagnosis of these genetic disorders. Or you may see abnormal splicing. That is very rare. That is only observed in the patient samples, not in controls. And that's another type of good evidence we're looking for in the RNA-seq data. So to begin with, I'll use 3 vignettes to help you illustrate, to help you understand what are we actually looking at when we employ RNA-seq to improve the genetic diagnosis of Mendelian disorders. The first one is the most straightforward, where we use RNA-seq to clarify the pathogenicity of variants identified from whole genome sequencing. This is a 23-year-old male with intellectual disability, epilepsy, and lissencephaly. We did whole genome sequencing and found a de novo variant in the gene DCX. That's responsible for lissencephaly. So that's a good phenotypic match. But the challenge here is that this DNA variant is deep intronic. It's 4 kb into the intron. And there are no other evidence to tell us whether this is a pathogenic change or not. Computational prediction from Splice AI says that there is a very low score. So there is some indication, but it's really not confident in whether this is pathogenic or not. We needed RNA sequencing. So we performed RNA sequencing on patient specimen. Now we can see that there is indeed a cryptic splice point here. So we're looking at the intron. There shouldn't be any junction. But in the patient, you're seeing that there are a lot of junction, there are a lot of sequencing reads. That's abnormal. That's going to lead to frame shift and loss of function of this gene. So that's helpful for interpreting this variant as a likely pathogenic variant. And next is the second example where RNA-seq is used in a more sophisticated way to help with DNA interpretation. This is a 6-week-old male with seizures, metabolic abnormalities, consistent with cobalamin pathway defect. And the patient is responsive to B12 treatment. Trio exome sequencing was performed, but it was negative. Then we got fibroblast RNA from this patient and performed RNA-seq, and we saw decreased expression of the MMA-CHC gene as an outlier. So that is consistent with the cobalamin pathway defect. That seems to be a promising lead. However, when you re-examine the exome data, we cannot see any variant in the MMA-CHC gene. Then we did whole genome sequencing and found a de novo variant in another gene. This is HCFC1, and it was not found in the exome because the region happens to be covered poorly. So why is that? Why is that different gene explaining the cobalamin pathway defect? It turns out that this HCFC1 gene is the upstream transcriptional co-regulator of the MMA-CHC. So the de novo mutation in that gene is leading to the downstream effect. This is reported in many papers, including this one, where signature is shown for the transcriptome data. And you expect to see these downstream genes, their expressions should change in a significant manner. This is consistent with our RNA-seq data. So that's further confirmation that this is a valid diagnosis. So in this case, we show that RNA-seq is utilized to illustrate a pathway outlier, pathway defect, suggesting a diagnosis. This final case is perhaps the most complicated example where we use RNA-seq to refine molecular mechanism of a disease. This is a 2-year-old female who had severe developmental delay, intractable epilepsy, cortical visual impairment, ASD, dysphagia, failure to thrive. We did whole genome sequencing and found 2 sets of candidate diagnosis. The first one is compound heterozygous variant in the gene WWOX. That is consistent with the seizure phenotype, and that's pretty straightforward. The second candidate is a de novo variant, but it's a VUS in the gene GRIA4. This gene is known to cause neurodevelopmental phenotype. So that could be a contributor to the patient's presentation. However, this is only a VUS. We don't know if this is pathogenic or not. And again, this is a deep intronic variant. So now the question is in this GRIA4 variant. We first looked at the known disease mechanism for GRIA4. GRIA4 is only known to cause disease in autosomal dominant gain-of-function mechanism. And this is slightly different from the other GRIA genes in the same family. The others can cause disease in both dominant and recessive manners. And then focusing on this variant, computational prediction splice AI says that there is a high probability that there's going to be some abnormality. And this is where the abnormal sites are predicted. The prediction says there should be a cryptic exon that is in frame. Right? So the consequence is that there are some additional amino acids included in this gene that could lead to gain of function, right? So that's suspicious. Then we did RNA-seq, and there is indeed some abnormality, but this is not exactly the same as what is predicted from the splice AI algorithm. There are many different possibilities. Overall, we see a decreased expression if you look at the number of reads compared to the controls. So this is possibly caused from the NMD, non-suspicious decay. So the read frame is not exactly in frame. That's different from the prediction. So now we know the consequence is possibly loss of function. That's inconsistent with the gain of function known disease mechanism. So we can say that at least based on what is currently known, this is not favoring the gene as a good candidate for a secondary diagnosis. So this is the most complicated use case of using RNA-seq involving understanding of the disease mechanism. This is becoming more and more relevant when you start to consider therapy, because if you design a therapeutic option, you want to know that you're at least treating the patient to the right direction. So the first case, as I said, is probably the most common use where there is a VUS and we just use RNA-seq to see if there's abnormal splicing or expression. The second case illustrates the pathway change. We are seeing this in a very small fraction of cases, but perhaps in the future we'll see more and more as we improve our understanding of this technology. And as I said, the last case, the disease mechanism is important when you start to consider therapeutic options. So that is RNA-seq used in a translational research setting. But what about clinically? There hasn't been a lot of clinical labs offering RNA-seq as a clinical test yet. So we started doing this. We started launching RNA-seq as a clinical test in the CAP-CLIA environment in the UDM. And this is work done several talented individuals in the lab where we conducted a validation of clinical RNA-seq in the UDM, the Undiagnosed Diseases Network. We picked 2 sample types, skin fibroblasts and whole blood. And we used pipelines as depicted here. And this is conducted in our CLIA lab, MGML, medical genetics and multi-omics laboratory. So there are several challenges when you start to do this clinically. One of them is there isn't really an established benchmark, which is needed if you want to validate any molecular testing. So we actually spend effort on creating a benchmark. We call it a provisional benchmark. This is a cell line HG002 that is being used frequently from the Genome Metabolic Consortium. And we gathered lots of sequencing data, short read, long read, and created a consensus junction companion, and then use that as a benchmark to compare with the data that we got from our validation. And as you can see, we get high recall and precision. So this is one of the requirements that a CAP-CLIA inspector will want to see if you want to validate NGS test as a clinical option. Then another challenge is you have to have enough interesting, positive, clinically positive samples to include in the validation. Because this test is not available, so it's very hard to obtain high quality clinical data with abnormal RNA findings. We are lucky to be in the UDN, and we were able to find 20 fibroblasts and 20 blood samples as positive collection that represent a wide range of abnormal splicing, including exon skipping, cryptic exon, cryptic donor, receptor, acceptor, etc., etc. And there are many genes of different challenges. Some of the genes are known to have a reduced expression that is at a larger scale. Some others are more challenging. They may have a more subtle reduction of gene expression. So we included all of those to challenge our pipeline. This is what we got from the initial clinical validation. And you can focus on this last column where we show the sensitivity of detection of the abnormality in high percentage. 95% of the diagnostic findings are identified from fibroblasts and in blood. And if you want to break them down and look at how many of them are detected from abnormal expression, from abnormal splicing, the data are here. So that is good, but on the other hand, if you want to have high sensitivity, you are also getting a lot of other false positive results. So at this very high performance setting, we are getting hundreds or thousands of artifacts at the same time. So that means every time you review a RNA-seq case clinically, you have to look at hundreds of findings, which is not feasible in a clinical environment. So we have to tweak the parameters so that we have a balanced sensitivity and positive predictive value. And then we have this alternative results where we have a reduced but still encouraging sensitivity, but then we have a much better, much lower false positive rate. You just are going to review dozens or small number of variants every patient in order to get to that diagnosis. We call that the agnostic analysis mode. So what this tells us is we now have a proposed strategy to review clinical RNA-seq. We should combine this agnostic analysis together with the targeted analysis, where we by default do this agnostic analysis for every case, where you look at them and you are confident in identifying the more easier diagnostic findings. You may miss some of the more challenging findings. And then you wanted to look at the DNA data so that they will provide you some of the hint where those challenging diagnostic variants are, and you can analyze it in a targeted mode, and that will reach a higher clinical sensitivity. So this is what we currently suggest as the way to analyze RNA-seq. So those are the basic understanding of using RNA-seq to help disease diagnosis and making it a clinical test. But looking into the future, how can we make RNA-seq better? As we know, RNA-seq is dependent on the inherent nature of RNA, which is there's tissue-specific expression, right? So we know that we are sequencing blood, fibroblasts most of the time. So that doesn't match to the disease tissue for many patients. Say a patient may come in with a seizure phenotype, and ideally brain is the ideal material, but you are sequencing blood and fibroblast. So we are not always getting the most appropriate material. And this study showed that if you are sequencing the clinically accessible sample, there are a large fraction of genes that you cannot activate, or you cannot detect in those materials. So if you focus on these regions, there are about 30–40% of the genes that are just not expressed in blood, fibroblasts, or muscle. So to tackle this problem, there can be multiple ways. I'm going to show all the three here, starting from CRISPR activation. So this is a paper published earlier this year in American Journal of Human Genetics, where they utilized the mechanism of CRISPR activation. So if you know of a disease gene that you want to activate, you can design a guide RNA towards the promoter of the region and deliver this mechanism into the cell so that this will activate gene expression of that inadequately expressed gene. They showed that this is achievable for MPZ and SPAST, two of the neurological disease genes. They can activate in fibroblast. So as they're showing their data, this is what you get when you sequence fibroblast directly. If you look at the numbers, those are very low numbers. The expression rate is low. But after CRISPR activation, the gene expression is improved, is increased a lot. So what about some alternative ways to address the same problem? One of the approach that we took is to simply sequence the sample deeper, where the expression is low. You just need to spend more sequencing reagents and reach a higher depth. So we know that a regular sequencing depth is at about 50 million reads. So here we sequenced to 350 million reads, and here we sequenced to almost 3,000 million reads. And you can see that we can detect the gene reasonably in this deeper RNA-seq data. The other way to achieve this is to make IPS and differentiate that into the appropriate cell type, right? So we actually have an approach to generate induced neuron in a rapid and low-cost manner, and this is the result from that protocol. You can see the gene is very well expressed. Very importantly, you'll see that there is the additional exon here, where this exon 1 is not expressed in blood, is not expressed in the fibroblast, and this is also in the CRISPR-A method. So this represents a tissue-specific transcript that is only activated in induced neuron and in the CRISPR-A. If you are selecting the right promoter, this is promising more diagnosis possibilities if a disease variant is in this intron 1. So going back to the first way we proposed, if you just sequence deeper, this is work led by Sen Zhang in the lab, where we sequenced clinically accessible samples, blood, fibroblast, and IPS and lymphoblastoid cell lines at very high depths. Again, the current clinical depth is on the left, about 50 million reads, and we sequence all the way to 3,000 million reads. And we show encouraging data that there are many more genes. Now we can detect them. We can detect outlier with very good significance. So there is indeed value if you can sequence deeper. We are able to do this because we've been working on another sequencing platform, Ultima Genomics, that offer low-cost sequencing compared to Illumina. The second approach that I just showed, which involves differentiating into neuron cells, is described in this paper that was just published about a month ago from our lab on the American Journal of Human Genetics. We show that we can take patient fibroblasts and do transdifferentiation, which means that you don't go through IPS, but you directly transdifferentiate that into induced neuron cells. And we show that this method is very robust. We did it in a large number of UDN samples with high success rate. This method is very effective. We found over 25% diagnostic yield with some of the genes in the neuron-specific, some of the diagnosis in the neuron-specific genes that you couldn't have achieved if you sequence only the fibroblast. And this method is low cost. That's ideal for clinical laboratory implementation. The cost is lower than $400 for the cell culture reagents. And there is shorter turnaround time compared to the alternative where you go through the IPS intermediate step. Actually just presenting this in the American Journal of Human Genetics Journal Club 2 days ago in Hong Kong. We're just arriving here. And if you're interested, you can Google this, and the recording is online where we talk in more detail about this protocol. So this is one of the cases that I illustrated in that paper. I wanted to present this, and I will come back to this case a little bit later when we discuss the therapeutic options. This is a 4-year-old male with congenital iris hyperplasia, developmental delay, hypotonia, ataxia, and mild dysmorphic features. There is an intronic de novo variant in the gene ATPR1 responsible for Gillespie syndrome that is consistent with the phenotype. This variant is -17 into the intron, and this is the VUS. So we needed to do RNA-seq to clarify the significance, but the gene expression is very low in the fibroblast. We made induced neuron, and that activated the gene expression by 12-fold, so we can get more accurate interpretation from the data, and we indeed show that there is abnormal splicing through the induced neuron RNA-seq. So remember this case, and I'll come back to this a little bit later. Now we're continuing to work on improving this protocol to adopt other sample types. I just told you that we can take fibroblasts and convert them into induced neurons, and now this version 2 protocol is amenable to blood, which is much more clinically accessible. Right? We can take patient blood and transdifferentiate that into neurons, and you see the performance of this protocol is very high if you compare the neuron-specific genes. Their expression in the induced neuron versus the blood, their expression is activated in a very high proportion of genes, over 90% or maybe over 90% of the disease genes related to neurological disorders are very highly expressed in this protocol. So now I'm going to present one more sophisticated case that we got interesting data from RNA-seq using this improved version 2 protocol. This is a 3-year-old male with autism, developmental delay, scoliosis, developmental regression, progressive high myopia, and spinal ganglion neuroblastoma. We did whole genome sequencing and found a de novo variant in the SOX5 gene. This is +5 intronic variant, VUS, right? We wanted to do RNA-seq to see what is the consequence of splicing. Splice AI predicted a reasonable probability that there is going to be abnormal splicing. And this is where the mutation is. And the prediction from Splice AI is that both the donor and the acceptor in the canonical transcript are going to be destroyed. So there is going to be exon skipping based on the prediction. And if there is exon skipping, the consequence is downstream frame shift because that exon is an auto-framed exon, and then there will be a loss of function result. This gene is not well expressed, it's very lowly expressed in the blood, right? So we need to activate the gene expression by making it into induced neuron. We did that, and the gene is now expressed reasonably in the patient versus the controls. And we did RNA-seq. You can see that there are several junctions suggesting the skipping of the exon, the skipping of this exon. Right? This is where the mutation is. This exon is skipped, but we detected two different sets of junctions. So we don't know which one is the majority of the consequence. Both of the numbers are very low, and they are similar in their intensity. And the numbers are low, possibly because of NMD. There is an abnormal splicing event that got degraded, so we cannot see what happened. Then in order to understand the precise mechanism here, we treated the cell with cyclohexamine. This is a chemical that can be put into the cell to suppress NMD, right? So if you add CHX into the cell line, you expect there is very low rate or almost no NMD. So we sequence this RNA data, we can see that among the two possible junctions, this one is activated, or this one is preserved much more. So this is the actual the majority of the abnormal splicing, right? So by adding CHX, we now know that this splicing variant resulted in skipping of this junction that lead to the final disease phenotype in the patient. And these are some of the more detailed data that you will see from data analysis, which I'm not going to show in detail. Yes. So to wrap up on the diagnostic part, I will have a small quiz here. So I always give this question when I teach RNA sequencing to residents, to fellows. Which of the following analytical scheme is commonly used in the clinical interpretation of diagnostic transcriptome sequencing data? Abnormal expression, abnormal junction, monoallelic expression, or all of the above. This is very straightforward. The answer to the fellows are all of the above, right? I talked about all the three, so they are all commonly used. But why do I put this question here? Well, this is a very straightforward question. What I actually wanted to ask is this more philosophical version of the question: which of the following is the most important component to you when you are doing this clinical interpretation in your role of providing patient care to the patient in question, right? So I will now argue that the answer can be different depending on your role. So if you're a physician carrying the patient, I may say that you are more likely to be interested in looking at abnormal finding of gene expression, because this is the most straightforward answer to you to say that if the expression is low, that suggests loss of function. If the expression is high, that suggests gain of function. It is more actionable to you. If you get abnormal splicing result, that may still be a VUS, right? You don't know what does it mean to the patient. However, if you are doing data analysis and your goal is to identify the molecular abnormal finding, you may be slightly more interested in this abnormal junction. Why is this? This is because even though I just said the ultimate result that you're looking for is this outlier expression, but that is very difficult to identify because of the a lot of the times because of the noisy data that we obtain from the transcriptome sequencing. So a lot of the times the data is too noisy for you to see a clean outlier expression. So you have to resort to the junction that is often a more direct indicator of the presence of an abnormality. Okay, now we are going to switch gear and talk about RNA-based therapy. So what I'm going to talk about today are focusing on this therapeutic modality, antisense oligonucleotides. This is a technology that has gained much popularity over the past few years. And this first slide illustrates some of the basic concepts. There are two ways to use ASO to treat disease mutation. So this method on the left is where you modify the oligonucleotides. They are usually around 20 nucleotide long in length. They are just like a primer that you use for your PCR reaction, even though they are not, but you can think about that, have a perception of the scale of it in that way. They're very easily synthesized, so that enable very accessible screening for us in the laboratory. If you modify the nucleotide in a certain way, you can design this ASO to target the disease mutation allele, and this will trigger downstream RNase degradation, and that will just get rid of the mutation allele. So if your goal is to reduce the expression of perhaps some dominant or gain of function mutation allele, this is the way to go. So this second method is where you design an ASO again to target a mutation site, but now you chemically modify it in a different way. And this will not result in RNase degradation. So this allele will not be degraded. However, this will lead to steric hindrance. So this ASO will just sit there and it will block the splicing machinery that is going to come here. So if there is for example, deep intronic variant that is creating a pathogenic cryptic splice site, your therapeutic goal is to suppress that, right? So you can design ASO that's like this, right? That will suppress the abnormal splicing and divert them to the normal splicing. So keep these two methods in mind, and we will discuss the utility of both of them with some examples later. ASO has gained a lot of popularity because of the very successful use case over the past few years in rare disease treatment, including this one, perhaps the most famous one, where Timothy Yu's group from the Boston Children's Hospital developed personalized targeted ASO to treat a patient with Batten disease. And that has received promising consequence clinically. And that has also approved FDA in a very rapid manner, resulting in patient benefit. So again, we're coming back to this case that I presented to you earlier. Remember, there is a disease variant, -17 intronic, and the disease mechanism is this abnormal splicing, right? So now we want to target this pathogenic variant. The abnormal splicing resulted in inclusion of 15 more nucleotides into the exon. So that is an in-frame change in the exon. And now we wanted to block this abnormal cryptic splice site and divert that back to the normal, push it back to the normal splicing. So we designed ASOs targeting this mutation site, right? And we have to design several different versions to tell the region so that we will screen them and select the more effective ones. And here are the chemical modifications that we have to provide to make this ASO to function as a stereohindrance ASO. We designed some PCR, ddPCR assays to demonstrate efficacy of this treatment. So remember now our goal is to suppress the disease allele, the allele with this 15 nucleotide extra inclusion. And this is represented as the red portion in all of these graphs. And each of the column here represent a different ASO. So compared to the controls, you wanted to see that the red portion got suppressed. So we see some of the very promising one like this ASO4, right? And you see almost a full reduction of this disease allele proportion. And on the left here, this is the same data represented in a different way, where we show the ratio of the wild-type allele over the mutant allele. So you expect to see a highly performing ASO should present you with a large value like this ASO4. And we did this in neural stem cells and in fibroblasts, and we got a good result on this ASO4. So that's a good initial lead. And then next, we want to focus on the ASO4 region and made some additional fine-tuning. We changed the length of the ASO a little bit and moved them to the left, to the right a little bit, and did some more screening. And eventually, we get some of the more promising optimized ASO that's demonstrating the highest performance in suppressing the mutation allele. We did RNA-seq on the samples treated with the most promising ASOs. And compared to the controls, our goal is to reduce this abnormal splicing portion. And you can see that some of these ASOs are really achieving promising effect. So that's the first case. The second case is where we use ASO in the other mechanism to elicit RNase mediated degradation. So remember, this is applicable to patients where we have a mutation that we just wanted to get rid of the mutant allele. Now we have a patient coming in as monozygotic twins. They both have de novo variant in the SPTAN1 gene, and their phenotypes are very severe epilepsy, cortical visual impairment, etc. So this mutation is in-frame duplication that result in insertion of a few extra amino acids. And the patient came in pretty soon after they got their molecular diagnosis in September 2022. So what does the gene do? SPTAN1 is a structural protein that encodes spectrin. Spectrins, there are the alpha version of the spectrin and the beta version of the spectrin. They can form tetramers, and they are important for the structure of, for the anchoring of many of the membrane proteins that's critical for neuron development. And this is possibly the mechanism that explains the disease presentation in these patients. And in particular, for this disease variant, previous research has shown that there is possibly a dominant negative mechanism where if you stain the alpha spectrum and the beta spectrum, they are actually forming aggregation, and as is shown in this staining figure, and that is not observed in controls. They are more in a diverse, diffuse manner if you look at a control cell line, but they're aggregated in the patient. And now our goal is to get rid of this disease allele and reduce the level of aggregation. Now, as I told you, the method that we selected is this RNase-H-dependent ASO that is designed to target the disease mutation, and the goal is to degrade it. We had an initial trial on designing an ASO targeting this mutant allele. However, it's not successful. Why is this? This is because the region of this disease mutation is actually repetitive, it's complicated. So on the top here, you see this is a reference sequence. This is in a healthy, the sequence of every healthy individual. There are already 2 repeats in the region, but in the patient, the disease mutation is addition of a 3rd copy. So this is a short repetitive sequence that made it very hard to target with a short ASO. We had to design ASO, but that does not distinguish the mutant allele and the wild-type allele because of the repetitive nature of the region. And also the ASO is probably forming some dimers, thus reducing the efficacy. And we see there is no selective suppression of the disease allele, and there is no effective reduction of this of either the disease or the wild-type allele. So we cannot do this by just targeting the mutation. Now we start thinking of alternative methods, right? If our goal is to just degrade that disease allele, we don't need to target the mutation. We can target other SNPs that are linked to that disease allele. So we did whole genome sequencing, and we faced all the SNP variants in the region, in the SPTN1 gene. And we selected the region that are more amenable for ASO development, such as having a reasonable GC content. And in the end, we came down to a short list of SNPs. This is where the mutation is. There are a small number of exonic SNPs, and there are some more intronic SNPs. They are all phased to the allele with the mutation. So we designed ASOs by focusing on some of the promising targets, and this is still work in progress. And we have some exciting preliminary results demonstrating allele specificity. Right? So you can focus on these ASO results in color, and you can see that the disease allele is actually suppressed to a higher degree compared to the wild-type allele. So now we stain the patient cell line again. This is a staining of the patient immune stem cell. And remember, the disease phenotype is aggregation, and we wanted to get rid of the aggregation. So we see, we don't see, we still see some of the residual aggregates in the cell lines treated with the ASOs, but we can already see some promising trend where the aggregation size and their number, their intensity are reduced in the cell lines being treated with the ASO. So those are just 2 representative projects that's going on in the lab. We have other ASO projects going on in the lab that are utilizing other gene regulation modalities. And these are some of the ways you can employ to activate gene expression. This is needed when the disease mechanism is haploinsufficiency, right? If the patient is having disease because they don't have enough of a gene, of a protein, now our goal is to increase their gene expression. You can either use an ASO to target the antisense transcript. And it is known that if you target the antisense, the sense transcript may have increase in expression. Or you may identify poison exons. These are exons that are naturally occurring in every one of us, but they are present in low fraction, and they are called poison exon because they result in a frame shift and they get degraded to MD in the end. But it's been shown that if you target these poison exons, you can actually boost expression of the normal allele. So that's the second approach. The third approach is involving translation and the concept of upstream open reading frames. So some of the genes, they have multiple open reading frames. And if you look upstream of the canonical translation start site, if there is an alternative open reading frame, and you may design ASO to block that, and you may have expected consequence of gene expression increase if you successfully blocked the upstream open reading frame. So some other projects going on in the lab, they are focusing on all of these different modalities, and I will share more as we proceed more into these alternative projects. So to recap, today I presented to you our progress using RNA-seq for Mendelian disorder diagnosis and therapeutics. And I wanted to say is very dynamic journey. And now we are moving into the multi-omics field, looking at RNA, especially the therapeutic side. This is always what we wanted when we started doing genetic diagnosis. The patient come in not just for a diagnosis, they really are looking for a therapeutic option. And we are so glad that we are very close to even implementing some of the clinical trials that may treat these devastating diseases. With that, I want to thank everyone who contributed to the work that I presented. This is my lab. I've done most of the work. In particular, I've shown a picture of Sheng Lan, of Sen and Jeff in many different slides. Really appreciate this is really an exciting team to be working with. And also showing pictures in the department of the training program, our residents and fellows. And yeah, I'm really glad to be showing the work here and sharing our experience. Now I'm happy to take questions.